Okay, so good morning, everyone. I think we can start with this today's TMS. So welcome, everybody. Today, I have the pleasure to introduce you Hannah Ford, that is a student of biological sciences at the University of Manchester, and is a placement student in the bone biopathology lab in the University of L'Aquila, and led by Professor Hannah Teti, and is also part of the TMS committee for the Biotech Journal Club. So today, Hannah presents us this exciting news regarding the transmembrane transport in inorganic colloidal cell mimics. So please, Hannah, the floor is yours.
Okay, thank you for the introduction. I'll just share my screen. Okay. Yeah. Can you see?
Yes.
Okay. So, as Michaela said, the paper that I will be sharing with you is called Transmembrane Transport in Inorganic Colloidal Cell Mimics. So first, I just wanted to show a bit of some news articles that talked about this paper because it is so exciting, and it's a very new paper. It came out in September of this month. So yes, there's some very nice articles talking about it. And also this YouTube video is very nice. I can leave a link if anyone is interested to watch talking about this paper. So firstly, I'll show you this video, which gives you an idea of what this is about. So you can see by this title that it's these artificial cell mimics eating E. coli bacteria. So this cell has a pump inside it and a pore, which allows bacteria to be taken up. You can see it goes inside. And this will remain inside the bacteria as long as the size of the bacteria is the same size as the pore in the cell. So it won't escape or anything. And this is to do with a chemical reaction which the pump performs, which drags material inside the cells. Okay, so first, as some background, what is synthetic biology? Because these are synthetic cells. So this is defined as the design and construction of new biological parts, devices, and systems, and the redesign of existing natural biological systems for useful purposes. So in terms of synthetic cells, there's two ways that these can be synthetically engineered. This is top-down and bottom-up. So top-down refers to taking an already living cell and modifying it, stripping it of things that maybe aren't necessary, or modifying the DNA using biological engineering. And bottom-up, which means starting from completely synthetic materials and making your own cell. And the cells in the paper that I'm going to discuss today were made using a bottom-up method. So for some background, for years now, scientists have been trying to create a completely artificial cell, and there have been successes in this field. This picture is from the first kind of case of complete gene expression within synthetic cells. So the cell on the left, you can see, is green because it's expressing green fluorescent protein, GFP. And on the right, this was an empty carrier. So obviously we've come a long way since this was 2003. And very recently, this paper discussed how cells were created which could grow and divide like, our living cells. This is a time course. You can see as time goes on, there's increased number of cells here. So, but until now, or until very recently, synthetic cells were not really shown to be able to perform a kind of active transport. So what is active transport? This is the movement of substances from an area of low concentration to an area of higher concentration, or against any kind of other opposing force, like an electrochemical gradient or something, through a pore using ATP. Now, this is active transport in living cells. So the question is, can synthetic cells perform active transport? Well, as you've just seen, they can, but only up until very recently. So this is the paper which showed the synthetic cells being able to perform a kind of modified active transport. So first, I will show you how they made the cells. It was quite a simple method, really. So they prepared an emulsion of oil in water, and then there was a process where oil droplets then self-inflated to become a membrane with an aqueous interior. Then they inserted the Phoretic pumps, which allow them to perform the active transport. And then these cells were placed in medium, which enabled them to perform the active transport. So I'll go through all of these steps in detail. So to prepare the emulsion, they used this oil precursor, which I'll just call TPM. They added this to some water and vigorously stirred. Now, this was to obtain a solution of hydrolyzed TPM monomers. So these are able to bond together, but first they just wanted complete monomers in a solution. Then this solution that they prepared was added to another solution containing ammonia and polystyrene spheres. These were negatively charged, which enabled them to be trapped at the oil-water interface of the TPM droplets. So the polystyrene spheres were a kind of seed for the growth of the oil droplets. So the TPM monomers began to form crosslinks between them around incorporating one polystyrene sphere each. So this polystyrene sphere is important for the creation of the pore, which I will discuss in a bit. So this was done under slow rotation for 18.5 hours to prevent the drops coming together or sedimenting. So this shows a kind of idea of the crosslinks between each of these monomers. So over time, as the emulsion ages, there's more crosslinks between each monomer. So after some time, the oil droplets were then kind of their required size for that step. So then there was a self-inflation process. Now this is a bit complicated, so I will try to explain, but the oil droplets were added to a sodium hydroxide solution. This then caused the oil droplets to inflate and become filled with an aqueous solution inside. This happens because the sodium hydroxide hydrolyzes this oil phase, which generates the hydrolyzed species which remain trapped inside the droplet. This then builds up an internal osmotic pressure, which is then counterbalanced by an external osmotic pressure, so the cells don't burst or shrink. This video shows this process, so I will just play it. First, there's a young emulsion with a high base concentration, so these have fewer crosslinks between the TPM, and you can see the aqueous moieties build up inside and then they stay these little cells. And so then this is an aged emulsion, so it has more crosslinks between this TPM network, and they're kind of more resistant to build up the aqueous moieties inside because the sodium hydroxide has a harder time hydrolyzing the TPM. But eventually they become cell-like. Okay. So in this figure from the paper, you can see, I'll talk about this graph first. So this on the x-axis shows the time in minutes that the emulsion was left to age, and on the y-axis shows the concentration of base in millimolars. And here you can see by these yellow spheres that if the emulsion was left to age too long or not a high enough concentration of base was used, the droplets remained solid spheres. They didn't inflate because there was too many crosslinks between the TPM. And here, by these pink crosses, you can see that if the emulsion was left to age for not long enough or too high a concentration of base was used, the droplets exploded. They went into solution. And the grey area shows the right conditions to form the inflated capsule, so the right balance of the number of crosslinks and the concentration of base. And also here you can see an idea for this osmotic pressure building up inside, which kind of was the basis for these cells to form. So now they have a cell with a bead in the membrane, which is important for the pore formation later. But first they wanted to fix the cells, so then they're kind of solid. They won't change size. So they used UV irradiation to do this. And this again shows a nice image for this. Also, this here shows what is inside of the cell, this aqueous moiety that's formed by the sodium hydroxide hydrolysis. So yes, starting with here, inflate the cells, and then they fixed the cells. So to create the pore, they simply washed the cell mimics in tetrahydrofuran, which selectively removed the polystyrene bead from the membrane. So they left with a cell or a vesicle with a hole in it. And here the different sized pores were created by using different sized polystyrene spheres. So next step was to get the pumps inside the cell. So the pumps were made of a solid photocatalyst, which is an iron oxide known as hematite. Here's a picture of hematite, but obviously they can't use a huge rock. So they used a microcube of hematite, which was just the right size to fit inside the pore and stay in the cells. These pumps were trapped inside the colloids by shaking the two species together, and then they isolated the capsules which took up the pump from those which didn't take up the pump. How does this pump work? Well, this substance has photoelectrochemical properties. So when the cell mimics were exposed to UVA or blue light, the hematite acts as a catalyst to decompose hydrogen peroxide into water and oxygen. This then created a chemical cloud around the cell, which attracted various cargo into the cell depending on the size of the pore. This sets up a concentration gradient between the outside and the inside of the cell, driving cargo across the pore in a process named diffusophoresis. This is a simplified explanation of this process so that it kind of makes somewhat sense here. But yes, this is pH and material dependent. So at pH below 6.5, material is repelled away from the pump. So if there was material inside the cell, it would then leave through the pore. And at pH above this, material is attracted to the hematite, so it will be pulled inside the cell. So to create the active transport solution, or the state to be able to perform active transport, the cell mimics were put in hydrogen peroxide solution and ammonium salt. And then there were some tracer particles added so that they could study the effects of the active transport. Then the blue light was shone on the cell mimics, and here you can see what happens here. So with the light on and at pH 9, material is pulled into the cells. Then with the light off, the cells are locked, so the material won't leave and no more material will be pulled inside. And then at pH 6, with light on, the material is expelled out of the cells. Here, yes, you can see that they can take up things that are not only spherical, but cylindrical in shape, as long as the circle part of this is the same size or smaller than the size of the pore. So there is a video here showing this. Okay, so first the ingestion. So there is a passive cell on the left which doesn't have the pump inside, and an active cell on the right which has the pump, so you can see the difference. So in the light at pH 9, the cargo is pulled inside the pore, and you can see it really sticks to the pump because the pump is pulling the material towards it. And when the light's off, the material is kind of more free inside the cell. And the light on, more material is pulled inside, the light off, stays inside. And it will keep going past the point where obviously the concentration of this material inside the cell is much higher than in the outside of the cell. So this is how this can be kind of classed as active transport. So it's still going. And yes, when the light is off, no material leaks out because they use the cargo, which is the same size as the pore. So it's very full now. So this shows that with the light off, there is no ability of the cargo to escape the cells, the cell mimics, I should say. And then reduce the pH to pH 6, and the cargo is then released with the light on. And this continues until it's completely empty. This larger circle is the pump inside. Okay, so go forward. Okay, so the future for these cells, or the potential future. In the paper, the researchers themselves talk about creating multicellular interaction systems, so building kind of big machines of these synthetic cells where some are tuned to take up cargo and some are tuned to release, so they can kind of communicate between them to form, I don't know, future applications. But personally, I think these could be the kind of blueprint for an exciting use as a drug delivery tool, because they can clearly, you know, selectively retain and release cargo, which maybe one day could be drugs. And also an antibacterial tool where perhaps in research settings where, you know, removing bacteria by other methods is less favourable, these can take up bacteria. So these could be used as an antibacterial tool. I also just wanted to talk about one kind of more real-life application of synthetic cells that's been seen. So this paper talked about synthetic cells that have been made to secrete insulin, which is very exciting. And so these cells that the researchers created were able to sense glucose concentration by a pH-tuned mechanism and respond to this by secreting insulin. But yes, this was a bit of an older paper, but shows a very exciting, you know, potential applications for this and the synthetic cells that I talked about today. And maybe if researchers kind of came together with their different aspects, their different ideas on the synthetic cells, like being able to divide and being able to perform active transport, then, you know, all the pieces could be put together to build a very exciting synthetic cell. And maybe one day we'll have a synthetic cell as complex as our own. So thank you for your attention.
Thank you, Hannah, for this exciting talk. It's very interesting, and I know it's a let's say very naive, but yeah, looks very nice. So, Professor Tati, please go ahead.
Yes, Hannah, I'm impressed by the beauty of the paper, the innovation, and also how you described it. Very professional. I think that the first junior club, we didn't practice that much, but yeah, to me it has been very good. I have a question actually. What I would see, it's a comment, what I would see as an easy way to exploit these artificial cells is making erythrocytes-like cells. Because the erythrocytes are very simple. They do not have organelles, just a cytoplasm
Yes.
30 more percent is made by hemoglobin. And if you have a membrane that is permeant to oxygen and CO2, I think that this could be a good application, and I would not see that far in the future.
Yeah.
I know that our people are working on changing the surface of erythrocytes to make them available for any type of patients, no matter the group, A, B, and 0, whatever. This could be another way how to solve the problem, because in this way you can make transfusions to everybody.
Yeah,
So,
definitely.
anything like that, or they want to go through to the complex?
I actually haven't really seen anything about that, like kind of taking the, taking advantage of how simple red blood cells are. But yeah, it could definitely surely be an interesting thing to look at. But yeah, the synthetic cells that I've been researching, they've kind of been trying to get them as complex as our, not other cells with nuclei containing cells, and kind of being able to replicate the processes that they perform rather than recreating a whole specific type of cell. I haven't really seen anything about that.
Yeah, I think that the challenge would be for that the shape of the cell.
Yes, yeah.
It cannot be round.
Although, sorry, I did see with the polystyrene spheres, if you use, you can use charged or non-charged polystyrene spheres, and if they had larger polystyrene spheres, then they could create the cells. I know it wouldn't be, the same composition, but it could be an idea of an application. They could use a larger sphere so that when the oil forms around the cells, they would have the biconcave disc shape. So it could be and to include 2 spheres, so then the cell forms in that kind of way. So it could be an application.
Okay, thank you. So this is really the science of the future.
Yes.
Thank you.
Thank you.
Okay, so I can see Professor Salamida with the hands up. So please, Professor.
Hello. So thank you. Okay, I will do a very naive question, because I'm not in this field. I'm very far from this field. But I found very interesting the technique to have these these synthetic cells doing something. So my question is, because this I'm asking, the question I'm asking is related to something I'm a bit sensible, because I have small children, and one of the problems is that they are always we are always using antibiotics for them. So when you speak about using these cells with bacteria, means that you can use these as in the future. Is clear, as a substitute of antibiotics or something like this. So instead of having something going around that is so in this way, basically you have, if I understand, you can also avoid the problem that the bacteria will become more resistant to the antibiotics.
Yes, I guess so. But the problem of using this inside the humans, which I just wouldn't know about, is that the cells can take up the bacteria, but the only problem, and they would remain trapped inside. The only problem would be potentially removing them from the body, removing the whole cell with the bacteria inside. But yes, as far as I'm aware, the cells, once they've taken up the bacteria, it would remain trapped inside. So it wouldn't kind of be affecting the body, but the problem would potentially be removing the cell from the body. That we don't know about how that could happen yet, but it's a very likely potential application. Yeah.
Comment on that. One can image, since we can image whatever we want.
Yeah.
Trap the antibiotics in these particles, attract the bacteria and kill the bacteria inside without spreading everywhere. Of course, this will not solve the problem of resistance, but would be safer for the patient.
Yeah, I hadn't thought of that actually, but yes, that's a very good point there.
Yeah, also, sorry if I can add, Marco Ponsetti also says, good for urinary tract infection, easy way to remove them. Maybe is a comment, nice comment. Yeah, also, yes, Professor, maybe it could be also nice to don't have the effect of the antibiotics in the other cells of the body if we trap inside this kind of.
Yes, this is true, because some of the antibiotics also influx the cells and they may damage that, especially in the mitochondria, of course, because the mitochondria are very similar to bacteria.
Yes.
Some of them are sensitive to antibiotics. So, in fact, some of antibiotics are also used as chemotherapeutics. So there is also, yes, this point is not only the resistance to the antibiotics, but also the damage.
Mm-hmm.
If you want to know more about that, you should invite Professor Perilli to give a lecture, because they are very skilled in this resistance to antibiotics.
Yes, sure.
Can I have always another comment in this
Sure,
direction? Comment is just speaking and thinking about things that does not exist. But I also can imagine that in the future, if the problem is to remove this cell or to control how this cell move in the body, you can in some way think about deep neural networks and also, let's say, learn, allow these cells or these things to know how to move and where to go.
Yes,
I
sure.
think something
Maybe,
like this in the future.
Yeah, maybe I don't know if Hannah reads something, but maybe embedded in these cells also include some, some
Instruction.
sensor, something like this. I don't know if Hannah reads something.
I think that the very final goal for these people in the very far future is to replace the neurons, because
yes,
could be very exciting.
They are trying everything they can to regenerate the neurons, but this is not possible.
Yes.
Because, at this stage, any idea is a good idea, and I find it very, very difficult because they are so specialized, so ingenious. But maybe for small damages, this can be used. For instance, the Parkinson's disease that is localized in the substantia nigra. This is a nucleus in the mesencephalus, so probably that could be a good target. But this is really a science of the far future.
Yeah.
I like to.
For you, not for me. I mean, you are young enough. Maybe in 20 years, you can be able to cure neural diseases.
Yeah, I like to think of it in the way that it took us billions of years to get us where we are, as complex as we are. So if the field of synthetic cells is kind of quite early, you can't rush it. But there is potential to kind of do it again, because we did it on our own. You know, we evolved to how complex we are. So maybe the field of synthetic cells, with all the technologies that we have, can get there one day, maybe.
Yeah, what I think is that maybe for this kind of stuff, I don't know if the top-down way should be easier, let's say. So if you take one cell and you make it free, like empty, and you use this like the, I don't know if you understand what I mean. So not create the synthetic cell from zero, but use the cells and make it empty and then use it for create new synthetic cells for the top.
Possibility, yeah. Why not? I mean, this is the time when people can have really ideas. Maybe 95% of these ideas will not be feasible, but for be aware that the progress in science, even at this time when we have a lot of technologies, it's slow. So it takes years with trials and errors, because maybe first attempts will be exciting, but not enough for progressing, and yeah, step by step. But I'm curious to know why Marco says that good for urinary tract infections.
To remove the bacteria, I guess.
Yeah, I think
No,
he's.
but why you don't tell us more, Marco?
Yeah, Marco, please.
Oh no, I was just thinking that basically if you could inject them in the circulation to cure urinary tract infections, you can make them. There are some antibiotics that do this, but you can make them go through the kidney directly and go in the bladder or the urinary tract, and then they could stay there for a while, collect all the bacteria, and then they would be removed the urine.
But there is an easier way to do that, Marco, because the bladder can be reached from outside through the urethra. So in this way, you don't disturb the kidney, because the antibiotics in the kidney, and I would say that these cells will be stopped in the kidney.
Yeah, that would be.
Go the other way around, as they do now for the chemotherapy for the bladder cancer. Influx the
catheter
through the urethra. Easier this way.
Yeah. Yeah. Anyway, yeah, that was just my point.
Yeah, it's a good idea.
Again, this is the time. I mean, it's nice. This type of
Discussion. Yeah.
You can just
imagine. Yeah.
Something that is impossible, but. You know, like Steve Jobs used to say, so you have to, to be creative.
Yeah, that's why when we found.
It's foolish in some ways. So it's.
That's why when we found this article, we were very excited to present you, because it's very naive, so you can imagine, and we thought about a lot of stuff, and we found a lot of very nice stuff regarding this.
Oh, we will establish the DISCUB laboratory of stupid ideas. I don't know, something like that.
Sure.
Next week we'll be very prepared. Yes, but it will be fun.
Yeah, and I don't know if anyone else have question. Otherwise, I can. Oriana, please.
I was wondering about the insulin and the synthetic cells delivering the insulin. Did these cells make insulin, or was it simply a carrier?
No, they couldn't make it themselves, so that's why, again, it's very naive. But so basically, vesicles containing insulin were pre-created and then inserted into a synthetic cell. So then when they sensed the change in pH, they could release the insulin by exocytosis. So they were quite short-lived. Obviously, they didn't have human applications, but if they did, they would kind of need to be modified to maybe have some kind of insulin producing mechanism, which would be fairly complicated, so that they could sustain themselves for longer, because the cells that they made could only maybe release insulin like 5 times, and then that was kind of all their insulin used up.
I was wondering, because I couldn't see the purpose without the cells producing the insulin themselves. Why would it be different to an insulin injection?
Yeah, it's just an application to kind of have potentially one day cells that can live for longer, and then will stay inside the body, so that any change in insulin, they can have a rapid thing. Say if someone was, had diabetes and was out and they forgot their insulin injection, they would have the cells inside them already to release the insulin as soon as the glucose drops.
But for this issue, you have 2 problems. One is to have the device, and the other is to have it regulated.
Yeah.
As Oriana said, there is no difference with injection if they are not regulated. So, and there are many groups that are working on that. You know why? Because if they succeed, they are going to make a lot of money.
Yeah.
It's such a frequent disease that, they are trying to make also artificial islets, or for instance, islets transplantation didn't work. Because the problem is that, I mean, the insulin technology is very advanced because they have very different types of insulins, and so to reduce the frequency and so on. But the problem is that insulin needs to be regulated. So it goes up when there is too much glucose in blood, and goes down when there is low glucose. And this is still being realized. But it's all material for your future, guys.
Yes.
Yeah.
Become excellent scientists, and maybe you will succeed.
Yeah, one more thing that I thought is that if we can put inside this kind of synthetic cells, the transcription, transduction machinery, should be very nice to have cells that maybe can at least divide themselves or survive for more. No, something like this. Do you read something about this?
Cells that can transcribe and translate or divide or both?
What kind of this? I don't know, one of the two or both. If you read something, if there is some studies that they try to, I don't know.
Yeah, so the cells in the picture that I showed early on in the PowerPoint were a top-down approach of synthetic cells. So they took bacterial cells and stripped them of all the non-essential genes so that they had, I think, 470 genes and were still able to divide and grow. So yes, they've done that, but not in a kind of bottom-up approach of entirely synthetic cells. And as far as transcription and translation, yes, they again, like the photo of with the GFP, the green fluorescent protein, they have made cells which were able to transcribe and translate proteins. But I didn't focus on that. I focused on the active transport just because it was quite complicated and maybe not as interesting to kind of look at. But yes, they have done that already.
Are very complicated things. So.
Yes.
Yeah, maybe it could be a good stuff to study to study how the cells work that make new cells, or for now, no, to try to, for example, throw all the non-essential gene and understand which one is very essential for the life of the cells, or understand how the cells were made at the beginning, so the ancestor of the cells, or something like this. Should be nice to use this kind of synthetic cells to most to study how the cells are for now.
Yeah.
What do you think?
Yeah, so they say that synthetic cells are kind of used for three purposes. So one is to kind of study the origin of life. So to recreate those ancestral cells and see, you know, what's essential and what's not essential and how they interact with things. Also, an application would be to kind of study biology without any or limited like kind of confounding variables. Like in living cells, there's so many processes going on, and maybe you want to study just one process singularly. So synthetic cells, because they're so simple and don't really have any other function to what you would engineer them to do, this is kind of an application. And then also to be used for future kind of technologies and things like this. So yes, definitely.
For this reason, I think that the erythrocytes are good target cells for this simplicity. I mean, it's difficult, the process of differentiation, but in the end, when they are mature, they're very simple. No organelle, just the shape, no dye, no regulations, just hemoglobin that binds the oxygen when it is abundant. And so I think that could be something, in my opinion, feasible in a reasonable amount of time.
Yes. Also, I agree completely. Yeah, for me, the most exciting part is to use them like an industrial, like in industry, no, to use this free new synthetic cells to put the ones that you told before, Hannah, to put one process, or start with one process and then again maybe add one more and then one more. So to try to, you know, for example, study one single pathway and then add maybe some lateral additional pathways to check if they work together or not, because it's difficult, as we did in our lab, for example, to study a pathway singularly, because there is a lot of factors that can influence at least also in vitro, not in vivo. It's difficult also in vitro to study one single pathway, to the regulation, the inhibitory part, all the stuff. I think could be very nice to use it in this way, like an empty industry for me.
Yes, definitely. I think bringing together the different processes that different scientists have focused on would be really nice. But again, the only problem might be is the use of different methods and different materials to make the cells with. But I mean, I assume that could be kind of resolved by finding a middle ground and things. But yes, it would be very nice.
Yeah, I don't know if anyone have some questions or comments. We can wait a few minutes, or I can ask one more. Okay, maybe I can go on with one more. For me, it's not very clear the limits between like vesicle and synthetic cells. When you can say, yeah, it's a synthetic cell, no a vesicle, or simply, carrier?
Yeah, I think that's maybe quite hard to define the difference, but I would assume that a synthetic cell is able to perform some of the functions that our cells can perform, like active transport. I don't believe that vesicles can perform active transport, so they are mimicking the processes that living cells, our cells, can perform. So they are mimicking active transport, they are mimicking translation, and vesicles can't do this. So yes, they do. They are, if you look at them simply, it's just a vesicle with a hole in and a pump, but it's the kind of looking at by the view of the processes that it can perform rather than what it's made of and what it looks like. You can call it a synthetic cell, I think.
Okay. So one more thing. Yeah, one more thing. Do you read something about using different kind of molecules, like lipids or something that could be more close to the normal cells to create this kind of synthetic cells instead of the TMP, right?
TPM, yeah.
TPM, sorry.
Yes, the cells that were engineered to release insulin were made of phospholipids. I think 5 different phospholipids were used. So they obviously had, the membrane was made with material that was more similar to our own cells. But I think it just depends on the purpose of the synthetic cell that is being made as to what material they use. So for these cells, I think they just wanted like a kind of a more sturdy material that can be fixed with the UV to stay solid. But yes, some people use phospholipids, or some people use the top-down cells. Obviously, use the membrane that was already there, maybe removing some membrane proteins. But yeah, different materials are used for different uses, kind of.
Okay, thank you. One more question from the chat. Marco Bonsetti says, for the chemists in the audience, so maybe not for us, do you think it would be an interesting approach to use for catalysis? I don't know if there is some chemists that want to reply, or because I think we are not able. Or Marco, maybe you can expand a little bit this question.
Yeah, I see a couple of chemists in the audience, but anyway, yeah, because one of the problems, for example, with the organocatalysis, which is a catalysis based on organic molecules like proline or derivatives of proline, is the concentration of the catalysts in the solution, basically, and having like these small pockets where you could put your catalyst inside could strongly increase the concentration of this of this catalyst, which could increase the reaction rates. This is something they do with other structures like fibrils or other kind of things. So I think these kind of synthetic cells could have a future also in that field. That's why I'm asking, but I don't know if there are any catalysts, organocatalysts in the audience.
Yeah. Yeah, I'm not the right person. I think also Anna is
I only
not.
know because my wife is, so.
Yeah, I think me and Anna, we are not the right person to, reply to your questions. But for sure, it could be nice to, maybe it could be a good stuff to improve. Why not? Again, all the stuff that you can think could be nice now because it's very naive field. So why not? Okay. So yeah, please, Ariana.
Kind of in follow-up to what Mark was saying, I was just wondering, the cells, the synthetic cells, can they take in more than one cargo, I suppose, or do they have to be specific to one thing?
They're only specific based on size. So as long as the cargo has the same size, or you can have spherical cargo and cube, what's the name? Cylindrical, as long as the kind of circle aspect is the same size or smaller than the pore. So they're only really selective if you are considering kind of keeping the cargo inside. That's only when they can't leave. But if you think of cargo that's smaller than the size of the pore, it will go in for sure because it's smaller than the size of the pore, but then it will also leave when the light is off. It won't remain trapped inside. So it's selective, yes, when you're thinking of trapping something inside based on size. But if something is the same size, you can use that. Or, you could have a mixture of the sizes of the pores. So you can have a mixture of the synthetic cells, some with smaller pores, some with bigger pores. If you wanted to take up, I don't know, materials of different sizes, if you had cells with pores size specific to those cargo, then yeah, they would be able to retain them inside.
And also, if I remember, we also read something about the square pore or
Yes.
different forms.
Yes, I forgot about that actually. You can have the polystyrene bead that's used to make the pore as different shapes. You don't have to have a bead, you can have a triangle or a square or things like a figure of 8 kind of thing. You can have different, you just need different polystyrene shapes to make the shape of the pore.
Mm-hmm.
The cells have multiple pores?
No, only one pore per cell because they use the right concentration of the polystyrene sphere so that it's highly unlikely for a cell to take up more than one bead. Or well, no, actually, the cells form on the bead. The cells form, so only kind of one cell per bead forms. But you could make them with multiple beads. Yeah, it would just be to do with changing the concentrations and the components of the solutions.
Mm-hmm.
I was just thinking in terms of catalysis, then, that if you had your synthetic cell able to uptake molecules and then they're confined with a catalyst that you've already included, then that could provide, as Marco was suggesting, a small concentrated pocket with the catalyst that might improve. But then if you have molecules of different sizes, do you need different size pores?
Yeah. Those
The short answer.
Yeah. Oriana is a chemist and catalyst chemist. No, but yeah, it could be a very nice idea.
Yeah.
Okay, let's wait just 1 minute for if you have more questions. Or Marco is yeah. Okay, so I think no more question here. So I would like to thank you for this very nice discussion and Anna for your very nice topic. And yeah, we can see in the next TMS. Thank you all.
Thank you.